All cells contain plasma membrane and cytoplasm. Whether we're talking about prokaryotic cells, eukaryotic cells, plant, animal, doesn't matter. All of them are going to contain plasma membrane and cytoplasm. But there are a few key characteristics that are different between prokaryotic and eukaryotic cells. Prokaryotic cells, that cytoplasm is going to contain water, inorganic and organic molecules, and enzymes. It is also going to contain a nucleoid region. Take a moment, see if you can remember what a nucleoid region does, or what does that mean. That nucleoid region is going to be the area where we house our DNA. Prokaryotic cells do not have a membrane-bound nucleus, but they do keep all of their DNA sort of bundled up in the corner to try and keep it organized. Eukaryotic cells, on the other hand, they have a cytoplasm, contains water, inorganic and organic molecules and enzymes. However, they have what are known as organelles, quite literally little organs found inside of the cell, each of which have a different function. The cytoplasm of a cell is going to be a semi-fluid solution, so it's kind of like a semi-solid, a little bit like jelly. It's going to be encased by the plasma membrane, and it's, like we said before, going to contain water, inorganic and organic molecules, and enzymes. The organelles of the cell each have a different function and are going to include the nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, and mitochondrion. We're going to spend a little bit of time going through each one of these. Non-membranous organelles are also going to perform very specific cellular functions, but the membrane-bound organelles include each of the following. All right, we're going to start with our organelles, starting off with our nucleus. The nucleus is going to be the command center of the cell. It's typically going to be found somewhat near the center of the cell. It is going to be separated from, contains almost all the genetic information of the cell. It's going to house the DNA of the cell, and it's going to be separated from the cytoplasm by a nuclear envelope. The nuclear envelope consists of a double-layer membrane. So here we have the plasma membrane on the outside. The membrane that surrounds the nucleus is actually a double bilipid layer. So it's twice as thick as that plasma membrane, that nuclear envelope. This is again to protect that DNA. We want to protect our hard copy DNA. We want to protect our hard copy information that tells every cell in our body how to be a cell. However, we need to be able to get that information into and out of that membrane, just like you house all of your information for this class inside of your textbook. It is protected. It is bound up inside of that textbook. It also is going to do you no good if you leave that information in the textbook, never crack it open, and never get the information. So what's going to happen is inside of the nucleus, we're going to copy down the information we need. We're going to write down the details. We're going to then, that information is then going to leave through the nuclear pores. Nuclear pores permit exchange between the nucleoplasm and the cytoplasm. The genetic information is going to be organized into chromosomes. Chromosomes are thread-like structures made out of DNA and associated proteins. And that thread-like DNA is going to condense down into visible structures just before our cellular division. So there you go. Genetic information is organized into chromosomes. These chromosomes contain chromatin. The nucleus contains chromatin. Chromatin is contains chromatin in a semi-fluid known as nucleoplasm. So cytoplasm is that jelly-like fluid in the cell. Nucleoplasm is the jelly-like fluid inside of the nucleus. Chromatin is going to condense into, contains nucleic acids and proteins, which can then condense down. Inside of that nucleus, if you can see right here, there we go. Right here we have what is known as a nucleolus. The nucleolus is composed of rRNA, that ribosomal RNA. This is going to produce the subunits of ribosomes. Does anybody remember what ribosomes are? All right, take a moment, write it down, figure out what are ribosomes, what is their function. These ribosomes are going to be produced inside the nucleolus of the nucleus. Here's another diagram of what that looks like. Here we have our we have our nucleus, our nucleolus, our nuclear membrane around the outside, nuclear pores found inside that nuclear membrane. And then we have rough endoplasmic reticulum on the outside, followed by smooth endoplasmic reticulum. And we're going to get into those next. Here is another picture or diagram of what DNA actually looks like when it's condensed. Condensed down here are individual chromosomes, DNA that has been condensed down for the use of mitosis, undergoing cellular division. So here's a little bit more information about our nucleus. As I've already mentioned, the nucleoplasm is made up of chromatin and the other contents of the nucleus. This is going to be that semi-fluid. The nucleolus is that specialized region within the nucleus that is involved in the production of ribosomal RNA, or the components that are going to make up ribosomes. Continuing with this, we do want to discuss what ribosomes are. Ribosomes, if you remember from earlier, are our protein factories. They are called the protein factories of the cell. They're going to be composed of a large subunit and a small subunit of our RNA. They're going to be located in a eukaryotic cell. They're going to be located in one of two locations. They're either going to be found on the rough ER or in the cytoplasm, and or both. In the case of a prokaryotic cell, we do not have rough ER, we only have cytoplasm, and that is where our ribosomes are going to be located. This is going to be the site of protein synthesis. The number of ribosomes found in the cell is going to vary depending on its function. So just for example, pancreatic cells have a lot more ribosomes than the average cell because they produce the protein secretions like insulin. If we're going to make a ton of proteins that are going to then be secreted out into the rest of the body, we need a lot of protein factories working on all cylinders at all times. So this is going to be the site of protein synthesis inside of the cell. Here is how those protein factories are set up. We are going to spend more time later on going over protein synthesis, DNA replication, but I do want to give us just a quick overview right now so we know how all of these components play a role. These protein factories work by starting off in the nucleus. The genetic information is going to be copied down as messenger RNA or mRNA. This is like you taking notes out of your textbook. You go to your textbook, you open up your textbook, you write down your notes, then you put away your textbook, you set that to the side, and you study using your notes. Or you bring your notes to class or whatever it is that you're going to do with your notes. You're going to copy down the most essential genetic information as mRNA. That mRNA is actually then going to leave the nucleus through those nuclear pores that we've already mentioned. The mRNA leaves the nucleus through those nuclear pores in the nuclear envelope, and it goes into the cytoplasm. In the cytoplasm, it's going to join up with a ribosome, either a ribosome attached to rough ER or ribosomes free-floating in the cytoplasm. Doesn't matter, they're going to have the same purpose, and that is to turn those instructions, those genetic instructions, into actual physical proteins. This, of course, happens at the ribosomes. The ribosomes are going to be the site of protein synthesis. They're going to read the genetic instructions from the mRNA. They're going to then produce a polypeptide strand. That polypeptide strand is then going to fold into a protein with the aid of chaperone proteins. They're going to help assist in the folding of that correct three-dimensional structure. As we've already mentioned, ribosomes can be located in one of two locations, either free-floating in the cytoplasm, or they may be found in the endoplasmic reticulum. The endoplasmic reticulum is a network of channels connected to the plasma membrane, the nuclear envelope, and certain organelles. So it is one great big long continuous membrane. You can kind of think of it a little bit like your skin. It's long and it's continuous. It wraps around, I don't know, this may be, maybe a little bit gross, but think about your face real quick. And specifically think about sort of the viscera and the holes in your face. So your skin, of course, starts up at the top of the head, and then it comes down over your eyes and then to your nose. But when we hit your nose, skin doesn't just stop being skin, but if you go into the nose, the nasal passages, you're going to find one continuous membrane going all the way back into your or your nasal cavity. Same with your mouth. You're going to find that your skin is going to wrap around your lips, go up into your gums, wrap around, and it's sort of all one great big continuous. Now, the types of tissue change slightly. Their purposes change. Their functions change depending on where they are in the body. But that outer covering of all of our outer covering is going to be all sort of continuous with each other. The membrane-bound organelles of the body are very similar. We have a double membrane that wraps around the nucleus. It's going to be continuous with the rough endoplasmic reticulum. It's going to be continuous with the smooth endoplasmic reticulum. It's going to be continuous with many other vesicles and organelles inside of the body. But each section, each area is going to have its own function and its own purpose. So we're going to look at endoplasmic reticulum. There are two types of endoplasmic reticulum, both with their separate roles and functions. The first type is rough ER. Rough ER is called such because it is studded with ribosomes. It has a very rough appearance. Because it is studded with ribosomes, what do you suppose its primary function is? That's right, its primary function is going to be protein synthesis. It's covered in protein factories. It's safe to bet that it's going to play an important role in making proteins. The other type of endoplasmic reticulum is what's known as smooth endoplasmic reticulum. It's called smooth ER because it does not have those ribosomes. This is going to actually be involved in the production of phospholipids and in detoxification. Smooth ER is incredibly important. It's used for synthesizing lipids. Here's an example of what that looks like. As I mentioned before, these membranes are sort of all continuous. So we have our rough ER, then we branch out from there and we're going to see our smooth ER, all one continuous body, but we differentiate it based on its appearance and function. All right, the next organelle we're going to look at is going to be the Golgi Complex. The Golgi Complex is a series of interconnected, flattened membranous sacs. This is going to be sort of our packaging center. We're going to pick up the proteins that were packaged in vesicles. We're going to transfer them to the Golgi Complex for processing and packaging. So once the rough ER makes these proteins, they're going to ship them off to the Golgi Complex. The Golgi Complex is going to sort and organize. This is the UPS center. It's going to figure out where these go. Do they need to go to another part of the cell? Do they need to go to another part of the body? They're going to be tagged and then sent on their way. Lipids are also going to be packaged and processed at the Golgi Complex. Lysosomes are our next organelle. Lysosomes contain about 40 different digestive enzymes that break down macromolecules, old granules, or old organelles, and invaders. Lysosomes, think of it as a little bit as the Lysol of the cell. It's used for breaking down, killing bacteria. It's going to be used for cleaning up the cell, getting rid of waste. Here is our Golgi Complex. It's going to be a series of these flattened saccules. What we're going to start off, we're going to receive vesicles coming off of the rough ER or the smooth ER. They're going to merge with this Golgi complex. They're then going to be processed, packaged, and sent on their way. Here's another diagram showing that. Here we have our proteins coming off of our ribosome-studded ER. They're going to be put into these transport vesicles, which will be carried to the Golgi complex. They're going to pass through, boom, 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 each one of these flattened saccules, and as they are being processed, they're going to be tagged to go wherever it is they need to go. So these they're going to either be packaged and sent to the surface of the cell, or they can be sent to lysosomes, which can be used to break down whatever it is that chemical is in there. Spend a little bit of time with this diagram. Make sure you can explain what each of these organelles are and what their purpose is and what's going on in this diagram. So spend a little bit of time. Anytime you see a nice big cartoon like this, it's meant to help you really understand and explain what's going on. So spend a little bit of time with that. Here is an overall diagram showing the entire process, start to finish. So like I said, spend a little bit of time. This is a great overview slide that will help you understand the different purposes and functions of lysosomes, how they break molecules down